I started studying medicine in the 90s, and regarding CSF flow, I learned this bulk flow model. So CSF is secreted in the choroid plexus, goes through the ventricular system to the subarachnoid space, and is reabsorbed at the granulationis of Paccioni and the nerve roots back into the systemic circulation. We already knew back then that this model was okay for simple things like aqueductal stenosis, but was not correct when it came to more complex things like normal pressure hydrocephalus, and complex models came up. In 2012, the group by Maiken Nedergaard added a very interesting concept to the conventional model named the glymphatic system. The name glymphatic system is derived from lymphatics that you have in the rest of your body, and we all know the brain does not have lymphatics, and from glial cells referring to the astrocyte end feet forming the blood-brain barrier. In this glymphatic model, the CSF in the subarachnoid space enters the space surrounding the arteries of the brain. There's an exchange between CSF and interstitial fluid. At the venous end of the interstitial fluid, more and more waste has been added to the interstitial fluid, and the interstitial fluid enters paravenous, not perivenous spaces and lymphatic vessels. Not only the glymphatic are different, but also the arteries in the brain have something special. In the human brain, we have about 600 kilometers of arteries. And in the video about stroke, I mentioned that the brain gets 20% of cardiac output. And Aristotle thought that this dense vascular network of the brain was meant to cool the blood, and he thought that all cognitive functions were located in the heart. So when you look at the arteries and veins in the body compared to the arteries and veins in the brain, they have been formed differently. In the body, the blood vessels are formed by vasculogenesis from mesoderm, which is regulated by a lot of complex genetics, and the blood vasculature forms a continuous loop. Then you have these lymphatic vessels intermixed, and they form an open-ended one-way system to get rid of all the waste. The blood vessels of the brain, the 600 kilometers of blood vessels in the brain, is mainly formed by angiogenesis. Angiogenesis is hypoxia driven, and VEGF plays a very important role. The first book I bought about clinical neuroanatomy was this very small book by Stephen Goldberg called Neuroanatomy Made Ridiculously Simple. And he described the arteries of the brain as a spider sitting in the center of the brain, and this spider was named Willis. And Willis was so ugly that he had no female counterpart, and the veins flee in the opposite direction. And that's because how the vascular system of the brain is formed. So when we talked about amyloid microangiopathy, we already talked about the cortical arteries that interact with the glial limiting membrane. And they are different from the lenticulostriatal arteries, because lenticulostriatal arteries are derived from the large vessel of the circle of Willis running in the subarachnoid space. So lenticulostriatal arteries are enveloped by a double sheet of leptomeninges, and I've outlined the leptomeninges in purple. and yellow is the glial limiting membrane and the astrocyte end feet. The cortical arteries are only covered by one layer of pia, so they don't have a real perivascular space. That's actually a subpial space. When you go to the level of the capillaries, the holes in the leptomeninges or in the pia become larger and larger, and at the level of the smallest capillaries you have more holes than pia. The veins of the brain arise in the capillary fields, and then the veins go to the deep cortical veins, superficial veins, and then eventually to the venous sinus. And this drawing of a human embryo at the end of gestational week 8 is already from 1921, and you can see that the arteries are running in the center and the veins superficial. And this is from the Human Virtual Embryo Project, where you can also see that the dural venous plexus is located very superficial. If you look at the telencephalon and the hindbrain. So the veins are also enveloped by some sparse pial cell islands, and the veins, they are not circumferential enveloped by a CSF space, but they have paravenous channels, so it's running next to the vein. And in this drawing you can also see it very nicely. So you have small holes in the leptomeninges, also purple in this drawing, at the side of the artery, and very large holes in the pial membrane at the level of the vein. So now we've discussed the arteries, the veins, and the basics of the glymphatic system. And in the next video, we can continue what happens at the level of the astrocyte end feet and how the waste is taken up, and also talk about the immune system, that you can also see the analogy in the lymphatic system of the body on this slide from Britannica. So I really hope you will stay tuned when we will continue with the blood-brain interface